next speaker is Professor Dipali Sharma, Professor, Department of Oncology, Sidney Kimmel Comprehensive Cancer Center at Johns Hopkins, USA. The topic of the talk is microbiota and cancer.
Hi everybody, good to see you all, and thank you for the organizers for giving us this opportunity to share our work with all of you here. So my lab has been working on various aspects of breast cancer for all these years, and today, this afternoon, I would like us to just sit back and discuss the role of microbiota in breast cancer. So I have no disclosures, and so if you think about diversity in human microbiota, so it's a growing field, and in the last maybe 10 years or so, we have come a far long way with the Human Genome Project, the completion of Human Genome Project by the NIH, and they actually looked at the microbiota of 5 body sites and showed the diversity and the richness of microbiota. And we know now that trillions of microbes live in and on our body. And with all these advancements over the years, but there's still some organs which were considered sterile, and breast was one of them. It was considered a sterile organ maybe 5 years ago, actually. And now the field has moved, and this is showing you the presence of bacterial DNA in human breast. This was some of the, here if you can see that, so samples were taken at different sites in breast tissue. I was trying to work the pointer actually. So the purple squares are the site of the tumor, and the blue round circles, if you can see those were the sites where the sample was taken for microbiota analysis. And as we can see here, and this is the stacked bar diagram. This looks very, very rich here in data. So every lane here is, every bar is a patient, and every box is the taxon, and the height of the box shows that the percentage abundance of that particular taxa. And here we can see that 2 cohorts were used here, samples from the Canadian cohort and the Irish cohort, and you can see that Bacillus and Acinetobacter were the most enriched microbes in Canadian cohort, whereas Staphylococcus was very enriched in Irish cohort. So I'm just showing you these data just to show the richness of the microbes in breast tissue itself. And now the field has moved so much that now we know that breast, not only it is not sterile, it actually has a rich microbe collection there. And here you can see in the first diagram here that actually if you compare melanoma, lung, ovary, GBM, pancreas, bone, and breast tissue actually has the highest load of bacteria. And as you can see below, these are, the first is the H&E slide, and then there is LPS staining, which is staining the bacterial load. And then there is the universal probe of 16S rRNA, again showing the presence of bacteria. So this data actually shows without doubt that breast tissue harbors rich microbiota. And if you look at the Shannon diversity, not only the number of bacteria is higher in breast tissue, as their diversity of the bacteria is also higher, and the percentage of bacterial abundance as well as the number of species that are present in breast tissue, breast cancer tissue, is highest as compared to the lung, GBM, and bone. So with that, I'm sure that everybody who's working in cancer area must have seen this particular diagram here, which is showing this, which is from a review from Hannan and Weinberg, and they've been actually revamping this. So we here see the hallmark of cancer, and all these hallmarks of cancer can actually be modulated by the intestinal microbiota. And now the field has moved when we know that there are organ-specific microbiota also, and all these microbes can actually impact various cancer-related processes, and they can help in cancer growth as well as metastatic progression. So here now I'm going to show you, so for the next few minutes, I'm going to show you 2 little stories from my lab. So the first one is that we looked at, so we did all this meta-analysis of all the existing metagenomic data, and then we analyzed breast cancer patient samples. We took normal and then breast tissue, normal breast tissue as well as the tissue from the cancer survivors. This is NAF is nipple aspirates, and then benign breast cancer and the malignant breast cancer tissue. And the idea was just to look for the oncogenic bacteria. So we sifted out some lists, and then we were looking at the bacteria, and then just to cut the long story short, we were focusing on Bacteria fragilis, which is actually, it's a very, very interesting bacteria. So Bacteria fragilis is an inhabitant of gut microbiota, and it's the non-toxin strains of this particular bacteria. They're normal commensals of gut, but the toxin-producing bacteria are actually pathogenic. And this bacteria can also modulate the growth of other strains of microbes. So it's like one bad neighbor, and that bad neighbor can, recruit more bad neighbors, kind of. We can kind of think about like that. It changes the biofilm in colon as well as wherever it is present. And B. fragilis produces this toxin, which is called BFT, Bacteria fragilis toxin. So when we identified this, we were pretty intrigued by this finding, because this is a colon microbe. What is it doing in breast cancer tissue? So first of all, we asked this question whether intraductal B. fragilis infection, can it actually modulate ductal epithelial cells? So we did this intraductal administration with B. fragilis, and first of all, we had one question whether it can actually survive there. And here we found out that not only B. fragilis survive in duct, it actually produces BFT, so it is viable and it is functioning. And then this is just showing you a snippet of the mammary glands of these mice that were harboring the infection of B. fragilis. And as you can see here, you can see the HNE, and if you can focus your attention on the first block of the pictures, the sham control, the normal ductal structure is there. And if you look at the ETBF, which is the toxic B. fragilis, enterotoxigenic B. fragilis, ETBF, you can see that all the ducts are actually filled with the cells, very potent hyperplasia there. And then we also used a mutant, the mutant of B. fragilis, which cannot produce BFT. So it's a non-pathogenic mutant. So with that infection, we found that again, the mutant did not affect the mammary gland epithelial cells, but then as you can go along the line of pictures that I'm showing you, you can see the Ki67 staining, the PCNA staining. So these cells that were filling up the ducts of breast tissue, they were very high in the Ki67 and PCNA expression, and we looked at CD45, CD3 staining also. And then this is just showing the quantitative representation. And I would like to discuss this particular cartoon here. You can see that there are normal cells, and then when the ductal hyperplasia takes place, these cells, which are very beautifully lining the ducts, they can actually start proliferating and filling up the ducts now. And as they fill the duct, we call them ductal carcinoma in situ. And then when they start invading out, we can call them IDC, which is invasive ductal carcinoma. So if you look at this cartoon now, and then focus on the data there that I'm showing you, where we actually scored the hyperplasia. So these mice were then given enteric infection of B. fragilis, and then these mice developed hyperplasia-like features within few weeks. It was pretty intriguing there, because they were healthy mice. The only infection that they had was the B. fragilis orally. And again, you can look at the pictures here, the control, ETBF, and the mutant. Control cells, beautiful ducts there. And with ETBF infection, we see this very potent hyperplasia in the second row, and the third row shows the mutant, which is not producing BFT, which is the toxin. So again, our next question was, now these were controlled experiment, of course. We were giving them the bacteria and just looking at the mammary gland hyperplasia. Now what happens if we give this bacteria to a model of spontaneous breast cancer? So this is a MMTV HER2. So this mice develops mammary gland hyperplasia, and then goes on to develop carcinoma within like 6 to 9 months. Every mice, all the mice get breast cancer. So here you can see in this particular slide, all the mice that were, the red line is ETBF infection. All these mice got a higher, they developed cancer earlier, so it was a reduced latency, and their load of breast cancer was actually very high. And they also developed lung and liver metastasis, as you can see here, in the lungs and the liver. So all the ETBF group developed higher load of breast cancer, as well as higher load of metastasis in lungs and liver. And now we ask this question that whether gut or ductal ETBF infection leads to metastatic progression on breast cancer. And here this is just a schematic of the experiment. These mice were given either ductal infection of ETBF or intraductal. So intraductal or gut infection. Essentially, our question is that if you give infection in the gut, can it actually travel to mammary gland? Can it travel to breast? So here, this particular first experiment is when the cancer cells were implanted intraductally, but the bacteria was given through the oral gavage. So it was essentially in the gut. And here we can see that red line is the ETBF group. Again, the tumor load was very high. And here in the next experiment, again, this was 41 cells which are very, very, I would say, highly aggressive model. Those cells were implanted intraductally, directly in the mammary glands, and then the bacteria was also given intraductally, which is again, directly through the nipple area. And again, we see the red line is the ETBF group, and we see that the higher load of mammary gland carcinoma here. And again, we see this higher liver as well as the lung metastasis. And then we asked whether gut colonization with ETBF stimulates early metastatic dissemination from mammary glands. And we see here again the red line is ETBF, and these mice actually in week 1 itself, when the primary tumor is not set up, these mice started showing signs of metastatic dissemination. So we found, as we can see in D, we see that lung and liver. At week 1, we see some in the ETBF group, we can see some signal there for metastatic lesions, and then week 2 and week 4, of course, in ETBF group, there was higher load of metastatic lesions in lung as well as liver. But if we compare that to the load of the tumor, primary tumor actually did not grow at a very high rate at that point of time, at week 1 and week 2, but the metastatic dissemination was already there. So it just shows that the cells were actually, they had this higher metastatic potential. So it was not that primary tumor grew so much that the metastatic cells were disseminating from the tumor itself, but the cells at the get-go were highly metastatic. And we looked at these pre-metastatic niches, and just showing you that, so this is a very busy slide, but the take-home point is that these group of mice that got ETBF infection, they primed lung and liver to, so that the metastatic dissemination can take place early on. And then they had a higher motility associated network. And these cells, as you can see here, these were the migration through these capillaries. And you can see here that these cells that were given ETBF, they migrated very high, at a very high rate. And of course, the toxin created higher tumor load in the BFT group, and these cells remembered. So I think the most beautiful thing that I can leave you here with is when this infection was given to mice, not only the tumor load increased, the metastatic dissemination increased. The other thing was that these cells were exposed to these bacteria for just 48 hours, and to the toxin maybe some in other experiment for 72 hours. But then the experiment took on for 3 generations of mice. So these cells actually, they acquired this molecular memory. So even after 12 weeks of living in mice, they remembered that they were exposed to BFT. So if somebody gets an infection with the pathogenic bacteria, their cells can actually remember, and then when other conditions are favorable, they can form tumor, and then they can go on to metastatic progression. So I'm going to now skip, because this is all the RNA-seq data, and all I'm showing is that, the enrichment of migration and all these invasion-associated genes, and we found the enrichment of Wnt, catenin, and Notch pathways. And again, as everybody does in molecular biology, we tested all these inhibitors in combination to see if we can overcome the effect of B. fragilis infection. And yes, bingo, we can. We can, combine this Notch and Vincristine pathway inhibitors, and we can then inhibit the effect of B. fragilis infection. And this is just showing you the cartoon, how it works. And I'm just gonna link this one thing for you so you can keep this in your thoughts. So we all might have heard that how oral diseases, and when you go to dentist, you must have heard that some of the oral diseases are actually linked with, say, cancer, maybe some cardiovascular problems and all. And here I'm showing you this data that F nucleatum, the first slide there, first picture there, every patient which had oral inhabitant of F nucleatum also had other microbes that all together they form a biofilm. And every breast cancer patient who had this bacteria in their oral cavity also had it in their breast tissue. So I don't know if you can follow the lines there, but the red lines are the presence of F nucleatum and Veillonella typica and the Veillonella dispar and parvola. And this next is where I want to leave you. So here we put intraductally, we put F nucleatum intraductally directly into the mammary glands, and we followed them. In 1 week, they all developed hypoplasia. And in 3 weeks, we actually had a low palpable tumor. It was pretty intriguing there because it's an oral microbe. And now, this is my last slide here, which is showing you in a nutshell what the bacteria can do. So these bacteria can be in the gut, but they can be impacting various organs. They can actually travel to different organs, riding these immune cells and the cancer cells. If there is cancer, they can ride the cancer cells floating around. They can ride the immune cells floating around. And these bacteria can actually affect drug metabolism, hormone regulation, immune modulation, anti-inflammatory cytokines, and all. And in dysbiosis stage, it is a reversible stage, but there are various other ways where dysbiosis can be tackled using all these different things that are listed there, using the spore-filled bacteria or bacteriophages. And now I think a very popular thing is fecal microbiota transplant, FMT. We're also looking at FMT and various other ways to modulate dysbiosis to achieve the eubiosis stage. And so we have a couple of clinical trials that are running, and we're analyzing all this data, sifting and trying to find who are the bad actors and trying to find the good actors, beneficial microbes as well as the oncogenic microbes. So I think I'm gonna leave you with that thought, thinking more about healthy diet and exercise and get to that eubiosis stage.
Thank you, Dipali.
Thank you so much. Excuse me. Oh, I'm sorry. This is my slide that I really need to show. This is the acknowledgement slide, my lab group at Johns Hopkins, and also the funding from various other organizations. Thank you so much.